Life as we know it hinges on the existence of proteins. These remarkable molecules are the workhorses of our cells, carrying out a dazzling array of functions that are essential for survival. From transporting oxygen in our blood to catalyzing biochemical reactions, proteins are involved in virtually every aspect of biological processes. Imagine proteins as microscopic machines, each with a unique shape and function. Their shapes are not arbitrary. They are intricately linked to their ability to interact with other molecules and perform their designated tasks. The process by which proteins fold into these precise three-dimensional structures is a fascinating and fundamental aspect of molecular biology. Understanding protein folding is crucial because even slight errors in this process can lead to misfolded proteins, which can clump together and cause diseases like Alzheimer's and Parkinson's. Therefore, unraveling the mysteries of protein folding holds immense promise for developing new treatments for these debilitating conditions. Proteins are made up of long chains of amino acids, like beads on a string. These chains are not static. They twist and turn, folding in on themselves in a process driven by complex interactions between the amino acids. The final folded structure, known as the protein's native state, is determined by the precise sequence of amino acids in the chain. This folding process is often compared to origami, the Japanese art of paper folding. Just as a single sheet of paper can be folded into countless shapes, a protein can adopt a wide range of conformations. However, only one specific conformation, the native state, is functionally active. The native state of a protein is not only the most stable conformation, but also the one that allows the protein to interact with other molecules with high specificity. This specificity is crucial for the protein to carry out its function, whether it's binding to a specific DNA sequence, catalyzing a chemical reaction, or transporting a molecule across a cell membrane. The instructions for protein folding are encoded in our genes, specifically within segments of DNA called exons. Exons are like individual blueprints that specify the amino acid sequence of different parts of a protein. During gene expression, these exons are transcribed into RNA and then translated into proteins. Interestingly, exons are not always contiguous in a gene. They can be separated by non-coding sequences called introns. This arrangement allows for a process called alternative splicing, where different combinations of exons can be joined together, resulting in the production of multiple protein isoforms from a single gene. This modular nature of exons raises intriguing questions about their role in protein evolution. Could exons represent ancient folding units that have been shuffled and recombined over evolutionary time to create new proteins with novel functions? Recent research suggests that this might indeed be the case. Section 4. Navigating the energy landscape of protein folding. To understand how proteins find their correct folded state, scientists use a concept called the energy landscape theory. Imagine a mountainous landscape with peaks and valleys. Each point on this landscape represents a different possible conformation of the protein, and the height of each point represents its energy level. The unfolded protein starts at a high energy state, like a ball rolling down a hill. As the protein folds, it explores different conformations, moving towards lower energy states. The native state, being the most stable conformation, corresponds to the lowest point on the energy landscape. However, the folding process is not always smooth sailing. The protein can get trapped in local energy minima, like a ball getting stuck in a small valley. To overcome these kinetic traps and reach the native state efficiently, proteins often rely on the assistance of molecular chaperones, which act like guides, helping them navigate the energy landscape and avoid misfolding. Section 5. Unlocking evolutionary secrets, exons as modular units. A recent study by researchers at Rice University and the University of Buenos Aires has provided compelling evidence to support the idea that exons could be ancient folding modules. The team used a combination of computational modeling and experimental techniques to investigate the relationship between exon boundaries and protein foldability. By analyzing a vast dataset of protein structures, the researchers discovered that exon boundaries tend to coincide with structural domains in proteins. Structural domains are independently folding units within a protein, often associated with specific functions. This finding suggests that exons might have served as building blocks for the evolution of multi-domain proteins. Furthermore, the researchers found that exons tend to encode sequences that are more likely to fold correctly on their own, even when isolated from the rest of the protein. This observation supports the notion that exons could have existed as independent folding units early in evolution, potentially giving rise to the first functional proteins.